So welcome to the Royal Society and the Leeuwenhoek Lecture for 2015. I have some housekeeping. You should switch off your mobile phones. There is no planned evacuations tonight, so if the bell rings, we've got to get out. You should go that way and the way you came in. And the event is being webcast and recorded. So I think that means for Jeff and for me, we've got to stand near this thing. Okay. So this Leeuwenhoek Lecture is given every 3 years now to recognize excellence in microbiology or microscopy, or both. And it's named for Antonie van Leeuwenhoek, who was a fellow of the Royal Society, elected in 1680. And he essentially, with his microscope, discovered bacteria. So that's the reason for his for naming the lecture for him. The lecture itself is supported by the estate of a man called George Gabb. George Gabb was not in the Royal Society. He wasn't a scientist. He was a chemist, and he was an I assumed he was a pharmacist, but I consider and he was an antiquarian and a collector, and he bequeathed also an etching of Leeuwenhoek to the Society. One of the things he did for the Society, which made his name a bit, was that he was shown a portrait of Joseph Priestley, which had been bequeathed to the Society, and he declared it a forgery. So that's his claim to fame. His real claim to fame is that he supports this lecture every 3 years now. So tonight's lecturer is Professor Jeff Errington, who graduated in Newcastle, then got his PhD in London, and then went to Oxford, where he eventually became Professor of Microbiology. He moved back to Newcastle in 2006 to become the director of the Institute for Cell and Molecular Biosciences, and he's now the director of the Center for Bacterial Cell Biology. Jeff's research is on the mechanisms involved in cell division and cell wall formation and their interactions, and in recent years he's studied cell mutants that have lost the ability to make cell walls. And I'm sure the importance of these studies will be a feature of his lecture tonight on bacterial cell walls, antibiotics, and the origins of life. It's my great pleasure to introduce Jeff Errington to give the 1915 Leeuwenhoek Lecture. Jeff.
Thank you very much, John, for that kind introduction to me, and also thanks very much to the Society for this wonderful accolade. Finally, I should thank Newcastle University, because they invested a lot in us. They saw the potential of bacterial cell biology, and a lot of the work that I'm going to be talking about to you tonight was done in this terrific building we're very proud of, called the Centre for Bacterial Cell Biology at Newcastle. So the lecture is going to be structured like this. I'm going to give you a brief introduction to microbiology, starting with Leeuwenhoek. I'm then going to tell you about bacterial structure and particularly focus on the importance of the cell wall. It's one of the reasons why it's really important is that it's a fantastic target for antibiotics, and so I'll tell you a little bit about how antibiotics that act on the cell wall work. And then, as John mentioned, I'm going to do a little bit of technical stuff towards the end of the lecture, in which I'll tell you about some of our recent research that, a little bit perversely, has been looking at bacterial cells that have no cell wall. These are called L forms, and about some of their really amazing properties. And then at the end, I'll talk about the implications for the origins of life, and you'll see how origins of life came into the research that we've been doing as a result of this work on L forms. So I want to begin really by stating something pretty obvious. Bacteria are extremely small. And I was looking around for something to scale them against, and I found that the £2 coin was very suitable. And then I realized that the 1997 version of this is actually very special. It's dedicated, for those of you who didn't realize this, and I didn't, it's special because it's dedicated to to scientific discovery. And on the edge of this is this little quote which came from Isaac Newton, which is very special to many of the fellows of the Royal Society. And what Newton said was, if I've seen further, it's by standing on the shoulders of giants. So I thought it was very appropriate to use that. And conveniently, this £2 coin is almost exactly 1,000 times wider than a typical human cell, which you see here. And so that means that you could fit about the best part of 1 million human cells on the surface of a £2 coin. So human cells are very small. But then if you actually look at a bacterial cell in the same slide, you'll see that bacteria are even smaller, or much smaller than human cells. And you could probably fit several thousand of these bacterial cells into one human cell. So this is about as good as it gets using light microscopy. You really see very, very little in terms of bacterial structure from light microscopy. You need to go to things like electron microscopy, which I'll mention later, to really blow these cells up. And then what I thought I'd mention, again, just to illustrate the problem that we're up against looking at bacterial cells, is that if you were to place a point green light within this cell, like this pointer actually, because we're actually very close to the wavelength of light. So this is the wavelength of green light on a similar kind of scale. What you actually see is a fuzzy blob of light using light microscopy, simply for the properties of the light. And so that means that the resolution of bacterial structure by light microscopy is very challenging. But as I hope to convince you, we're learning a lot about the ultrastructure of bacterial cells now through this bacterial cell biology field. So we have to begin with Antonie van Leeuwenhoek. As John mentioned, he's really the father of microbiology. He was the first person to realize that there were microscopic animals, if you like, or organisms. And this is one of his microscopes, this tiny microscope. We'll contrast it with some of the microscopes that we use now soon. And these are some illustrations that he put into some of the letters that he sent to the Royal Society. And the microbiologists out there will recognize the major morphological classes of bacteria. There are basically 2 or maybe 3 morphologically distinct classes. There are rod-shaped things like this. There are spherical things like these tiny things that we would call cocci. There are helical things. And one of the other things that he noticed, which was amazing, is that he noticed that bacteria could actually swim. So this cell is actually moving. And in fact, they can swim in a directed manner. And I'll show you a little bit later one of the proteins that's involved in actually detecting chemical gradients and which cells use to direct their swimming. So actually, remarkably little happened if you look at the history of microbiology over the next 200 years. And it was really not until the end of the 19th century that microbiologists really connected infectious disease to particular kinds of bacteria. And one of the heroes of this period was Hans Christian Gram. And towards the end of the 19th century, he invented this staining method called the Gram stain, which is actually such a fantastic stain that it's still used to this day by clinical microbiologists. And so so it distinguishes between gram-positive bacteria, which are blue or purple, and gram-negative bacteria, which through the stain actually appear pink. And this is important because it turns out, and Gram couldn't possibly have known this, that the difference in Gram staining is actually to do with the structure of the cell surface, which is different in gram-positive bacteria from gram-negative bacteria. And these organisms, the gram-positives and gram-negatives, probably diverged from each other something like 1 billion years ago. So it's a very important distinction which Gram discovered and which is still used. And so again, we can see 2 different kinds of bacteria, we can see their shapes, but really that was all we had to go on with in terms of microbial structure for many decades. Now, in the middle of the last century, the problem of resolution was really solved by the advent of electron microscopy. And so this is a modern, typical electron microscope from Newcastle. And basically, there's an electron gun here. There's a specimen chamber here. The electrons are fired at the specimen, and then an image is collected at the bottom of the machine here, and it can be displayed nowadays on a computer display. And this solved the resolution problem, but it's at a cost. And the cost comes from the fact that electrons are very easily scattered, even by water molecules. And so to get good images through of biological specimens with a bioelectron microscopy, you have to dehydrate them, you have to take the water out, and you have to do several other things. For example, you have to fix the cells with horrible chemicals that make lots of chemical crosslinks to stop them being destroyed when you dehydrate them. And then afterwards, you have to embed them in plastic. Then you have to section them with a diamond knife to get sections which are thin enough to be able to be visualized by electron microscopy. And then finally, these specimens are actually relatively transparent, and so you need to stain them. And typically, you stain them with something like uranium, very heavy metal atoms, to actually be able to resolve these or to see anything in these specimens. So when you do all of this, and so there are lots of potential for artifacts here. What you can see are really basically 4 distinct structures. There's this sort of fluffy white stuff in the middle, which is where the DNA is. There's a granular material called the cytoplasm, which is mainly ribosomes and the protein synthetic apparatus. Then there's this very prominent cell wall structure on the outside. And just inside that is a cell membrane, which isn't really very visible here. This is a semi-permeable membrane, which separates the inside of the cell from the outside. So this gave the impression of a rather featureless kind of cell structure. And that stayed with microbiologists or biologists really for several decades. It gave an impression of simplicity and lack of compartmentalization or regional specialization, which I think has really held the field back until recently. So all of this changed in the 1990s, and Roger Chen was one of the heroes of this. He won the Nobel Prize with his colleagues for realizing the potential of taking protein perversely from a jellyfish, this kind of jellyfish here, Equoria. And these are wonderful creatures. If you go to the tropics and you look in the sea at night, you can see these jellyfish glowing. And so they generate their own light, and one of the proteins that they use to generate light is this protein called green fluorescent protein. And what Chen and his colleagues did was to harness this as a tool for actually imaging all sorts of things in biological systems. So in bacteria, what we do is we take a gene which is interesting to us, which might be involved in cell wall synthesis or cell division, and we use genetic engineering methods to take the gene which came originally from the jellyfish and actually fuse it to our gene of interest. What then happens is that this generates a hybrid protein which hopefully is functional, which is produced the same as the protein which is made by our gene of interest, except that it has this green tag on it. And this gives us that pinpoint of green light, which I mentioned in the early slide, which enables us to follow using modern fluorescence microscopes where this protein goes inside the cell. And so we can do this to all of the proteins in bacterial cells, and we can ask where do they go in the cell. So this is in contrast to van Leeuwenhoek's early microscope. This is one of the microscopes that we have now in the CVCB. It's a commercial microscope. And what's amazing about the way this technology is developing now is that these microscopes are starting to overcome some of the limitations of light microscopy due to the wavelength of light. And so this gives us something like a 2-fold improvement in resolution over the best fluorescence microscopes that used to be available. And you see the complexity of this system. Here's the microscope, but then there are all these other add-ons, lasers and cameras, and then the whole thing gets connected to a computer which collects the digital images and where the images are processed. So I thought in the next few slides, I would show you some examples of what we've been able to do, what we and many other labs have been able to do in terms of understanding bacterial ultrastructure. And so these are some people from my lab who went to their favorite proteins and got me some images to look at. So I might need to put the lights down a little bit just for the next few slides. So this is a very simple experiment. Here we've taken this green fluorescent protein and we've hooked it up to a protein sequence that takes it to the membrane. And so this basically stains the membrane of the cell. And so you can see this beautiful green outline of these B subtilis cells. And you'll also see that in some of them, they're actually initiating a division, a cell division event, which will eventually lead to the formation of 2 cells. This one's just about completed division, so there are now 2 cells. And you see other cells are in different stages of division. So this is all very simple. What we've done in the next few slides is to replace that membrane stain with an actual red dye, which stains the membrane. So we can see the outline of the cell, much as you saw in the previous slide. But then we've connected the green fluorescent protein up to a series of different cellular proteins. So in this example, we've connected it to a protein called HBSU, which is what you might call a chromosome organizing protein. So this protein basically binds to the chromosome in the cell, DNA material, and it localizes all over the chromosome. And so what you see is the outline of the chromosome in these cells. And what's interesting about these images is that you see these very often bilobed structures. So there are two blobs of green that are connected together in each of these cells. And we think these are chromosomes in the process of being replicated. And ultimately, these two blobs of green will resolve, and then the cell will divide in the middle to produce the two daughter cells, each of which has a copy of the intact copy of the chromosome. So this is another DNA binding protein, but this one is much more specific. It localizes in one very precise place on the chromosome, which is called REC, and this is the place where the replication of the bacterial chromosome initiates. And it always initiates at the same single site in bacterial chromosomes. And so these green spots that you see here actually correspond to initiation sites on the chromosome. And if you count these spots inside the cells, you can see that this cell has 4 spots, this cell has 4 spots, and this one has 8 spots. And what this tells you, this kind of experiment tells you, is that bacteria are actually amazingly able to reinitiate a round of DNA replication before the previous round is even finished. So they can kind of get ahead of the game. And one origin will turn into 2 origins, and then each of those 2 origins will turn into another 2 origins, giving you a total of 4. And the cell is able to kind of organize its chromosome with these multiple ongoing rounds of replication and somehow manage to stop everything from getting completely tangled, so that when the cell eventually divides, each of these cells will obtain a chromosome complement. So here's another kind of protein. This brings us back to that point I mentioned right at the beginning about the bacteria being able to direct their swimming. So this is a protein called a chemoreceptor. This is effectively the bacterial nose. So this is a protein that can actually detect chemical gradients. And what you see with this protein tagged to GFP is that the protein is directed to the cell poles. And you get these little spots of localization, 2 spots of localization often, at each of the cell poles. And so this is where the bacterial nose is. These proteins help the organism to figure out, to recognize chemical gradients and to respond to them so that they can swim in an appropriate direction. So now we're getting to some proteins that will feature later in the talk. And so this is one of my favorite proteins called FTSZ. And so this is basically the master regulator of cell division, and it's present in virtually all bacteria. So this is the protein that organizes the division machine and enables bacterial cells in general to divide. And so at this level of resolution, the FTSZ protein has a very interesting localization pattern. In these images, we're actually looking at effectively a cross-section through the cell. And you'll see that there are 4 cells here, and they're actually in different stages of division. This cell has an incipient division ongoing. This one's just starting to divide. And these 2 cells are kind of in the stages of not having initiated division yet, but they've already localized the division machinery in this FTSZ protein at their right site. And what you see if you're sharp-eyed, perhaps, and sitting in the front, is that in every cell there are 2 spots of FTSZ. That's because this is a ring structure, and you're looking down through the ring and seeing the two spots that represent the sides of the rings. And what you should be able to see is that the rings here are very widely spaced. The FTSZ protein is right at the periphery of the cell, whereas in these 2 cells that are dividing, the green spots move closer together. And that tells you that the FTSZ stays at the leading edge of the septum as the division machinery actually constricts. So you can actually follow division in real time by labeling proteins like this. So here's another of my favorite proteins, a protein called MREB. This is now a master regulator of cell shape. And it has yet a different localization pattern. This protein makes long filaments that are often appear to be helical and which wrap themselves around the long axis of the cell. And this protein is one of the key proteins that determines the shape of the cell. And so these cells are rod-shaped, and most rod-shaped bacteria actually acquire that shape through the action of this family of proteins, the MREB proteins. And for those of you who are interested in cytoskeleton, this is a structural homologue of human actin filaments, which are important in cell shape in eukaryotic cells or human cells. So this protein wraps itself around the long axis of the cell, and it actually controls the way that the cell wall machinery synthesizes new material. Okay, and then finally, I've switched for one slide from B. subtilis to E. coli, and that's because there's another class of really amazing proteins in bacteria that actually can shift their position on a remarkable time scale. So this is a MinD protein, and it's involved in determining the location where the division site is placed in many bacteria. And in E. coli, it does this remarkable oscillation. And so you see the protein starts in the middle, then it goes to the outside pole, then it goes back to the middle, and then back to the outside pole, and back again. And so it does this oscillation with a period of something like 50 seconds. Really remarkable behavior. A lot of people, physicists and mathematicians, are interested in trying to understand how this works. But basically what this protein does through this oscillation is figure out where the midpoint of the cell is. And it's one of the proteins that's really important in positioning that FDZ ring that I mentioned earlier right at the middle of the cell. I hope you noticed in those other cells, the Z ring is always right at the middle. And so there are very sophisticated mechanisms that are involved in positioning the Z ring in the middle, and this is one of them, at least, which is used by some bacteria. So that's kind of an introduction to where we've gone with microscopy from the very early crude light microscopy through to absolutely modern, up-to-date fluorescence microscopy with super resolution imaging. So what I'm going to explain in the next couple of slides is just where my particular research interests have been for a number of years, and that is mainly in these questions about cell shape and about the cell division cycle. And the reason why I think this is really amazing is shown if you look at a time-lapse image of my favorite organism, Bacillus subtilis, undergoing growth and division. And so this is greatly speeded up, of course, but these cells are going through a complete cell cycle in about 20 minutes. And so this is a whole series of probably 10 or so cell cycles. But if you look carefully at these images, what you'll see is that the cells are incredibly precise in the way they undergo division. They basically elongate, they maintain a constant diameter, they elongate, and when they've more or less doubled in length, they divide right in the middle to produce two baby cells. The two baby cells, both of the two baby cells then grow, double in length, and then divide in the middle. And they do this over and over and over with incredibly high fidelity, and it's very rare to see a cell make a mistake in division in these kinds of time lapses. So how can the cell organize its chromosome, its growth, its division, so that every single molecule in that newborn cell is completely duplicated 20 minutes later, and that the two baby cells are generated. So it's an amazingly complicated biological problem, but one which is, of course, really, really fascinating. So the bacterial cell wall turns out to be a key player in both growth and division, perhaps not surprisingly. So what is the cell wall? Well, in a sense, it's a tough, stretchy fabric that covers the whole cell surface. One analogy I like to use is of a kind of spring vest. So one of the reasons why you need the string vest on the outside here is that, as you can see, the cell cytoplasm is very concentrated. There are lots and lots of molecules inside, whereas the outside medium is very dilute. For those of you who did A-level biology, you'll realize that there's an osmotic problem because the water outside here wants to rush in and equilibrate with the high concentration of stuff inside. And so that generates a very high pressure, turgor pressure, pushing out on the membrane. And the only reason this cell doesn't swell up and burst is because it has this string vest covering its whole surface, which will constrain its growth. So the cell wall has pivotal roles in both cell growth and division. And if you imagine, coming back to the analogy, what the cell needs to do is to introduce new stitches into the vest so that it can extend it. And then when it's doubled in length, it has to then divide the string vest into 2 identical copies, all the time retaining this very highly charged cytoplasm intact. So the wall turns out to be the crucial target for our best antibiotics: penicillin, cephalosporins, vancomycin, and many other antibiotics that are still the foremost weapons that we have in combating infectious disease actually work on cell wall synthesis. The cell wall is also recognized by our innate immune systems. So when bacterial cells die, they release fragments of the cell wall, and our innate immune systems recognize those fragments and respond appropriately to the threat of infection. The wall has a very wide distribution, so it's thought that it was probably present in the last common ancestor of all bacteria. So all major lineages of bacteria have more or less the same cell wall structure. And so that suggests that the very first bacterial cell on the planet probably already had a cell wall. And so what I'll come back to at the end is what happened before that cell wall was actually invented. So the wall is an ideal target for antibiotics. It's essential for growth and viability. There's this internal osmotic pressure, which is very dangerous if wall integrity is breached, and I'll show you this in a minute. Importantly, there's no equivalent of the cell wall in humans. We have none of our cells have cell walls, and that means we can get selective toxicity. We can get chemical compounds chemical compounds which interfere with cell wall integrity will kill the bacterial cell, but we don't have to worry about any effect on human cells. So we kill the bacteria without killing the patient. And so these are still our best antibiotics. But there is, I think, the potential for new classes of antibiotics still targeted on cell wall synthesis, because there are still many elements of cell wall synthesis for which there are no clinically useful antibiotics right now. So this next movie is from my friend and colleague from Ken Gerdes, who developed this movie, I think, for a BBC documentary about a year ago. And what it shows you is a time-lapse image of E. coli cells. On the right are the control cells, which you'll see are growing very nicely. And on the left are cells that are sitting on a bed that's been impregnated with penicillin. Oops. And so as you see, these cells grow very nicely, just as the B. subtilis time-lapse that I showed you. But on the left, you'll see that there's all kinds of devastation occurring. And these cells are basically popping. I think we'll run that one more time, because this idea of mass destruction of bacterial cells, I think, is something that most people would like to see happening. So let's run that again. You can see, I think towards the end of the movies, there's a cell down here that actually shows this beautiful exploding event. I think it's this one. So there's a breach in the cell wall, and then basically the cell contents just simply explode from the cell. And that's because of this turgor pressure, and it shows you how important this structure is for bacterial cells. So we've been interested for a long time now in trying to discover and develop new antibiotics. And one of the targets we and others have been, pharmaceutical companies in fact, have been interested in, is this FDSZ protein that I mentioned earlier on. And so FDSZ, I'll give you a different perspective on the GFP fusion in this image. In this image, we've got a three-dimensional image of the cells, and we've actually tilted it so that instead of looking down through the rings, you can actually look along the cell. And now you see these beautiful ring structures that are being formed in virtually in every cell that's actually about to undergo division. So with colleagues at a spin-out company called Proliferation, I see there are one or two ex-members of Proliferation here, we succeeded in developing inhibitors of FDSZ, specific inhibitors of FDSZ, and actually showing that they had considerable potential as antibiotics. So this is the movie you've already seen. This is the movie of cells which have been treated with a specific inhibitor of the FDSZ protein. And what you'll see is they grow quite well for a while. They elongate, but they don't divide. Then eventually they become unstable. And if you look at the end of this cell, you'll see that, again, just like the penicillin-treated cells, they kind of explode at the tip. So we think that one of the things these compounds do, apart from blocking cell division, is they result in disturbance of the final division that occurred when the compound was added. And eventually these filaments become unstable, and you can see that all of the biomass here, all of the growth that occurred in the presence of the antibiotic is really futile because the cell is going to burst. So we were very excited about these compounds and invested a lot of time and effort in trying to develop them. And several years ago, we were successful in getting at least a proof of principle that we could find novel potential antibiotic molecules that would work on this new target FDS-Z. Again, it's associated with cell wall synthesis. And this is a paper I'm very proud of from several years ago now. And this is one of the compounds that Proliferation developed. And we were able to show that these compounds don't just kill Bacillus subtilis cells, which are non-pathogenic, but they also kill Staphylococcus and even methicillin-resistant Staphylococcus. And they'll do that in an animal model of infection. Sadly, these compounds are not yet in development. I think there's still quite a lot of interest in developing these molecules or other FGS-Z inhibitors, but so far, issues such as the development of resistance have stood in the way of actually getting these compounds into clinical trials. But I think it's an interesting proof of principle that there are still novel targets out there that could be exploited by people looking to develop new antibiotics. And of course, as everyone here knows, there is already a desperate need for new classes of antibiotic that we use to try to combat the insidious threat or insidious rise of resistance. So everything I've told you about so far has stressed how important the cell wall is. And you'll see that my lab has been working on cell wall synthesis, cell division, regulation of cell wall synthesis for many years now. And so a few years ago, we became interested in this really puzzling question about how bacteria can survive and maybe even thrive in the complete absence of a cell wall. And so the way in which we got into this problem was that there was a long literature of work on bacteria called L forms. And these L forms are also called cell wall deficient bacteria. So they were first described 80 or so years ago by Emmy Kleinenberger. And just to preempt a question that I often get asked about where the L came from, an L form, it actually stands for the Lister Institute in London, where Emmy Kleinenberger was working. So there's a huge literature on these L forms, mainly from clinical situations where people have been treated with often with beta-lactam penicillin-like antibiotics, which actually selects for organisms that can grow in the absence of a cell wall. So L forms can also be made in vitro apparently by treating cells with antibiotics or secreted enzymes that damage the wall. You have to retain them in special culture media to prevent them from bursting because of that turgor problem that I mentioned. And they've been implicated in many chronic or persistent diseases, but the problem is that the organisms are very slow growing and they're difficult to culture. And the evidence is often very weak. Most of the reports are individual case studies, and the controls are often either completely lacking or quite poor. And one of the big problems is that this is mainly from the pre-molecular era, when it was really quite difficult to study these organisms. And I did a recent PubMed search just to look for L forms, publications on L forms. And what you'll see is that there's a big peak of L form papers back in the 70s and 80s, but that since the sort of molecular era in the middle of the 1980s, the number of publications has kind of dwindled. And in fact, I think I would argue that it wasn't really until about 5 years ago when we could start to do whole genome sequencing, taking advantage of the new DNA sequencing methods, that we really started to have the tools to be able to look at the cell form question properly. So we became excited in trying to understand, using modern molecular genetics, what gave us, if you like, with these L forms. So the heroes of the story in my lab are Richard Daniel, just there, I think, and Mark Levy. Richard's a long-term associate of mine. Mark was a graduate student who wanted to stay on for a year to do a high-risk, high-reward kind of project before he went off to become a postdoc working on worms, I think. So they developed a tractable system for studying L-form biology using our favorite organism, Bacillus subtilis. So there's a technical warning here. So the next few slides will have a little bit of technical detail on. So I apologize to those of you who have no microbial or little biological background. So this shows you the method that we used to develop the L-form system. So the synthesis of cell wall has been well worked out over many decades. And the basic raw materials for cell wall synthesis are actually synthesized inside the cell membrane. And there are a series of enzymatic reactions which are well known, which are shown here, which lead to the formation of this small molecule called lipid 2, which is then flipped through the membrane to the outside, and it's then assembled into the existing cell wall to make these are basically new stitches that are being put into the cell wall fabric. So in V. subtilis, it turns out that the genes that encode 5 of these enzymes sit together in a little cluster, and they're co-regulated, so they're turned on and off together. And so we used genetic engineering to alter the regulation of these genes so that basically the synthesis of these 5 enzymes, the expression of these genes, is dependent on whether or not a simple sugar called xylose is present in the medium. If you put xylose in the medium, then these enzymes are all made, the genes are turned on, the enzymes are made, cell wall synthesis proceeds normally. But if you take away the xylose, then you shut down expression of all of these genes, and you have a multiple blockade in this pathway, which prevents the cells from improving or expanding their cell wall. So, cut a long story short, there was quite a lot of technical difficulty in getting this to work, but eventually we came up with a protocol which worked, which is shown here. We stripped the cell walls, we put in a lot of sugar as an osmolyte to prevent the cell from bursting due to its turgor. We have no xylose in the medium to prevent the cell wall synthesis through the route that I mentioned previously. For technical reasons, we had to put an antibiotic selection against the rods in there. There'd be lactam penicillin antibiotic. And what we found was that we needed to have at least one additional mutation. So in addition to shutting down the cell wall synthesis, so if you like, in addition to that mutation, we needed a second mutation to get the L forms to proliferate. With that mutation, we then got these things that look like classical L forms, so very, very different morphologically from these walled cells. But this very heterogeneous population of cells is typical of the sorts of L form organisms which have been reported in the literature over many decades. So, very early on in this project, there were 2 huge surprises, and this is the reason why we're still working on this problem very hard right now. The first one is that this FDSZ division machine that I've been talking about and telling you was a wonderful target perhaps for new antibiotics becomes completely dispensable in L forms. So here's the experiment that we published a few years ago now. We take this FDSZ gene, we engineer it so we can turn it on or turn it off. In normal cells, if we turn off the FDSZ gene, then we get this extension of the cell, they filament, and then they burst. And so that's completely lethal. But in the L-form background, it makes no difference at all to the growth of the cells, whether or not you turn the FDSZ gene on or turn it off. And the referees first forced us to go even further, and so we showed that we can completely delete the FDSZ gene, we can take it away completely from these cells, and it makes no difference whatsoever to the proliferation of L-forms. So the L-forms must be using some completely different mode of proliferation to the one that's used by virtually all of the bacteria on the planet in normal circumstances. So that raised an interesting questions about how the L-forms actually would proliferate. And here was the second surprise. And that is, again, I'm going to show you this movie. This is how normal walled cells grow. And this is what the L-forms, how the L-forms proliferate. So you'll see again that they're rather heterogeneous. There are very big ones. There are actually tiny L-forms. Some of them appear, this is sort of tubular material here, which you can't see very well. And when you look at the mode of proliferation, it's really, I still haven't found the right adjectives to describe this. I mean, basically the whole thing is quite a mess. It seems very haphazard and heterogeneous. In some cells, you see this spontaneous emergence of several small, tiny progeny cells. Sometimes you see a chain of cells being generated. But it's very rare to see cells that appear to be doing anything like binary fission. It's very difficult to see anything in these movies, so what I've done is to show you a couple of still sequences that illustrate some of the common events that occur in those movies. So one kind of event, you see this round L form growing in size, and then it starts to elaborate this tubular structure, which then grows and eventually breaks down into a chain of apparently connected progeny. So you see the emergence of 5 or 6 cells in a single event. And here's another larger L form. Again, it's more or less spherical. Then you start to see these pulsating changes in the cell surface, and eventually there's an eruption at several points on the cell of a series of small progeny cells. So they're very, very strange events occurring in these L forms. So we think these are proliferative events. You can follow these baby cells and watch them grow and undergo similar kinds of proliferative changes. So one of the, I'll tell you in a moment that we've been studying this mode of proliferation in a lot of detail over the last few years. But one of the big questions that we had was whether these very strange events that we've seen for B. subtilis would actually apply also to other bacteria. So in the last year or so, we've looked at a couple of other very diverse bacteria. Corynebacterium glutamicum is another gram-positive bacterium like Bacillus subtilis, but it comes from a completely different lineage of the gram-positive bacteria. And E. coli, of course, is a well-known gram-negative bacterium. It comes from that other completely different, the two groups of bacteria that were discovered by Gram 130 years ago. This comes from the other group. Okay, so there's 1 billion years of evolutionary divergence between these cells. But when you look at them, when you convert them to L forms using more or less the methods that I've just told you about, then you see that they look remarkably similar. So Corynebacterium actually grows even better than Bacillus in liquid, but again, it shows this very kind of haphazard blebbing and tubulation kind of process. E. coli is a little bit different. This is going on in liquid, as for the B. subtilis L forms. With the E. coli L forms, we've been unable to get them to grow in a completely liquid medium, so they're growing in a semi-solid agar medium here. But again, you're seeing rather similar proliferative events, given that these cells are partially immobilized. Again, it's a very haphazard, non-binary fission-like process that's being undergone in these cells. And one of the questions that we've been able to, I think, answer now, finally, relates to the question about how different these L forms are from, how to define actually L form bacteria. And so what we've been able to do, again in E. coli, just as we did for B. subtilis, is we've been able to, once we've generated these L forms, actually show that we can delete these key essential cell division genes, the FDZ gene and other cell division genes and other cell wall synthetic genes. Once you've turned these into L forms, you can completely dispense with all of those genes. So these are really remarkably different from walled cells, but it looks as if a similar kind of process can be followed in very diverse branches of the bacterial domain. Okay, so for the last few years, as I mentioned, we've been pursuing the sort of molecular basis for L form division, and these are some of the people who've really been playing prominent roles in the lab over several years now. And I won't tell you much about this because a lot of this work is now published, but we think we can identify several discrete steps in L form proliferation, and for each of these steps, we've been able to identify genes that B. subtilis uses to be able to carry out, to be able to proceed through those steps. And so on the next couple of slides, or in fact, maybe the next slide, I'm really just going to focus on one piece of work to do with the growth and deformation, which I think is the most informative piece of research that we've done on the L forms. And so I'm going to jump really to the model that we have now for how L forms proliferate. So we were very uneasy with this model. We published this about 2 years ago now. But it turned out that all of the genetic evidence that we had and all of the imaging information that we had pointed to what we thought was an unexpected model. We were anticipating to begin with that there would be some kind of protein-based machine that would bring about this division process, because there's always a protein machine in biology. It turned out, though, that at least the genetics, all of the genetics that we did told us that all you need to do to be able to generate an L form is to actually increase the rate of membrane synthesis. So you unbalance the normal regulatory link between expansion of this growth of the cytoplasm and growth of the surrounding cell membrane. And so it leads to a model that looks like this. You start with a spherical cell, which has just the right amount of membrane to cover the surface of the sphere. And then if you imagine that you have, well, what normally you would imagine would happen would be that the cell will regulate the synthesis of membrane so that as it grows, it will maintain just the right amount of membrane to cover the surface of the sphere. However, in the L forms, they actually make excess amounts of membrane. What happens then is, from simple geometry, the only way to lose the excess surface area is to change the shape of the cell. Because as soon as you, this object with this crazy shape requires much more surface, much more membrane to cover its surface than a sphere of the equivalent volume. So it's simple geometry that some of us or some of you would have learned at school. So the upshot of this is that if you make excess membrane, you actually drive deformation of the cell. And if you take that to its logical extension, you can even pinch off some of these lobes that are generated, and that generates a bunch of smaller cell progeny. And again, because of simple geometry, the collection of smaller cells that you generate, or the total surface area of these small cells, is greater than the surface area of a single cell of equivalent total volume. Okay, I hope that makes sense. But I'm going to try and convince you that this model could be right, by the end of the talk. So this is the model that we were forced to go to. It's an extremely simple mechanism. It doesn't require any kind of complex protein machinery. All you need to do, and this is again what the genetics tells you, is make more membrane, and the cells will spontaneously carry out this kind of proliferation. But you need to have the excess membrane synthesis. So now, those of you who've looked at my title carefully will be wondering where the origins of life starts to come into this. And so it turned out that when we started to look at started to think about this model for L-form proliferation, that we had to start reading about how membrane vesicles behave. And that actually led us into the story that I'm going to tell you about right now. So the next 2 or 3 slides are just going to tell you what current thinking is, as I understand it, for what the key steps were in the evolution of life on Earth. And I've drawn from a very nice chapter that Rich Losick wrote very, very recently and shared with me, called The Origin of Early Evolution of Life. And so the first key step in the evolution of life was the availability of the small precursor molecules like amino acids, nucleobases, and fatty acids, which are used to make the proteins and nucleic acids and membrane material that go to make cells. And so the idea was that there was a kind of prebiotic soup that contained these things in abundance, and that was where life eventually emerged. And I think modern thinking is that these kind of chemical building blocks could indeed have been available on the early Earth. And the source of those could either have been from outer space, because I think chemists can now, spectroscopists can now detect these kinds of small molecules in deep interstellar space, in deep interstellar dust. Or it's also possible that they could have been formed on the early Earth. And there were some amazing experiments carried out in relation to this hypothesis back in the 1950s. Stanley Miller did these experiments where he took you imagine the sort of gases that would have been around on the early Earth, things like methane and ammonium and hydrogen. He put those gases into a cylinder. He boiled water, so you had water vapor going through the cylinder. He then had an electric discharge. He ran the experiment for long periods of time, and then looked to see what kind of molecules fell out into the solution down here. And what he found in these experiments was, remarkably, you can generate from this kind of experiment a large proportion of the key ingredients that you need actually for life. So it seems as if this step in biology is probably reasonably well understood, at least in principle. The next step is to go from the soup to something that has a kind of information content. And so I think, again, a lot of scientists would agree that it's very plausible that the first information-containing molecules that were around on Earth, on which evolutionary processes could start to develop, was based around the so-called RNA world. And so the idea here is that critically, RNA is an information store. Like DNA, there's a sequence of bases which can encode information. But critically, unlike DNA, RNA can also fold into complex structures like this one. And in this state, these RNAs can have catalytic properties. So they can catalyze reactions that nowadays would probably be catalyzed by enzymes, by protein enzymes. But in this prebiotic world, the RNA, it's thought, would have been carrying out a lot of the early chemical reactions that were needed for life. And what's beautiful about this is the catalyst contains the information that's needed to encode itself. So the idea of this would be that DNA and proteins were simply later improvements, that this was a better storage molecule for information, and proteins were better catalysts. But of course, all of this is speculation, and probably you need a time machine to go back. But as I understand it, the aficionados of this field now recognize these two key steps. So the third step requires some kind of container, because for Darwinian selection to begin on those RNA molecules with their diverse sequences and their different catalytic properties, some would be better than others, for the catalysts to actually undergo any kind of selective enrichment, the RNA molecule and the products that it generates need to be retained in some kind of container. And it's now thought, again, plausible that the container was probably fatty acid or lipid based, because fatty acids and lipids will spontaneously assemble into membrane vesicles, and it's thought that molecules like fatty acids and lipids were probably, again, present in that prebiotic soup. So finally, this is where a lot of the thinking has moved to in the last few years, is the question about how this membrane container actually could have replicated. So it's all very well having a lipid vesicle with RNA molecules and small molecules inside it, but how can that vesicle proliferate to generate two vesicles and retain the products of RNA in there? And so this is Jack Szostak is one of the main proponents of this stage in understanding the origins of life right now. So it turns out, when you look at the literature, that the people who are really interested in the origins of life have concluded that it's in principle very simple to enable these vesicles to proliferate, and in fact, so the theory is actually quite well worked out, and the key parameters turn out to be increasing the surface area to volume ratio, and secondly, that the vesicle should be relatively impermeable, so that when you start to make the shape change, the water doesn't just simply rush in and equilibrate everything. But in principle, at least, in theory, you can drive these kinds of shape changes and even proliferation simply by increasing surface area to volume ratio. The experimental biophysicists have gone one step further. And so again, around about 2009, this paper, beautiful paper, was published. And there are a number of similar kinds of papers, but I particularly like this one. And so this is a simple in vitro experiment where they have taken phospholipid vesicles. So this is a simple single phospholipid chemical species which has been assembled spontaneously in the test tube into these spherical vesicles. What they then did, and these are very stable, they will sit there for many hours. What they then did was to start to feed these vesicles by adding fatty acids to the medium. And those fatty acids will intercalate into the lipid vesicle, increasing its surface area. So they can replicate the theoretical experiment that I showed you in the previous slide. And when you look at what happens to those vesicles, you see these amazing proliferative events. Here's one that's blown up. Here's a sort of tubulation kind of event that's occurring in some of these when there's a little bit of mechanical agitation. And what we became very excited about was the fact that these images look very reminiscent of the pictures that we saw for the alpha-1 proliferation. So we've become very excited by the sort of convergence of the top-down approach that we've been using to sort of strip out of the cell wall synthetic machinery, the division machinery from bacterial cells to revert them to some kind of primitive state, which is converged with the bottom-up approach that the origins of life groups have been using to try to assemble the sorts of structures that could have been used by primitive cells to proliferate. So I think it's turned out to be a very exciting area of interest for us. We think that the L forms provide a very interesting model system for future studies on the origins of life and how very primitive cells could have come about. And it's also interesting to speculate, just coming back to that point that I made earlier on, about the first bacterium already having a cell wall. Of course, you can imagine that before the cell wall was invented, then maybe the bacterial cells actually proliferated like the L forms that we're describing here, in which case the invention of the cell wall structure and synthesis could have been a defining moment in bacterial evolution, allowing bacteria to escape from their muddy puddle, where Darwin would have had them first evolve, to get out onto the planet and colonize the whole of the planet and generate all of the diversity and the abundance of bacterial forms that we see today. So that really brings me to towards the end of the talk now. I've got one final or a couple of slides to go now. First of all, it's just a summary and a perspective of what I've tried to tell you about. The key thing is although bacteria are very small, they're incredibly sophisticated in structure and function. I hope I've shown you that many bacterial proteins, perhaps most proteins, have very specific addresses that they go to and can find in the cell. They go there to perform the very sophisticated functions that they carry out. Of all the structures in bacteria, the cell wall, in a sense, is really, in one sense, the most important. It's a crucial structure for viability of the cells. It's important for shape and division, and a superb target for antibiotics, perhaps for future antibiotics as well. Remarkably, though, bacteria can escape from cell wall damage by switching to this L form state. And they can do this with only 1 or 2 mutations, and this seems to be conserved across perhaps the whole of the bacterial phylogeny. And we think that the studies of L forms have wide-ranging implications. We can imagine using them as model systems for studying all synthesis and division, basic science, because we can delete many of the genes that we would normally want to study. There's a lot of interest in using these kinds of approaches to generate artificial cells, which might have lots of biotechnological applications. There's still a huge question, clinical question, about whether L forms are important for recurrent or persistent infections. And finally, there's this really interesting question about the potential of using L forms as models for the origins of life. And that brings me, I think, to the last slide. I have to acknowledge that none of the work that I've talked about tonight was actually done by me. I've been exceptionally lucky to have had a whole string of very talented and dedicated researchers come through my lab over the last 25 or 30 years. This is the present group outside our favorite building. These are the people. These are the current crop of students and postdocs whose shoulders I stand on. I'm privileged to stand on in presenting the science that I've told you about tonight. Thank you.
We'll have some questions.
Sure.
Yeah. Questions? Yes. There's someone in the second row up, a lady in a red jumper. Ah, it's Marianne.
Jeff,
Marianne.
can you hear me? Yeah.
Yes.
Have you told us how these L forms or indeed the Urbacteria deal with osmotic pressure?
No, I haven't.
How do they?
I think there are 2 answers to that. One is that you can, one possibility is they actually only live in niches where there is some kind of osmotic support. And so you can imagine in certain clinical situations, there could well be, serum. It could well be that in fact, we know that some L forms can survive in blood and serum. The second possibility is that you can actually train L forms to grow on lower and lower osmotic media. We still don't understand exactly how that works, but we have L forms that are growing without any added sucrose to normal microbial culture media. So you can't train them without too much difficulty to actually grow. And I don't understand the biophysics of that at all yet.
In one of your slides, it looked as if there was a gene product involved in the sort of nipping off of the small L form. Was that a correct interpretation?
No, what you mean in the schematic slide? We look for mutations that block L form growth specifically.
Right.
And so we characterized one mutation in a lot of detail. And it turns out to be involved in membrane synthesis again. It's involved in the formation of branched chain fatty acids.
Right.
And these branched chain fatty acids are responsible for controlling the membrane fluidity. So we think that this mutation reduces membrane fluidity, and then the scission event that you need to separate those cells occurs less efficiently if you have only a higher membrane fluidity. So we don't think there's a specific scission. I mean, we're very much hoping to get proteins that might be involved in scission, but always we only found things affecting membrane synthesis.
In the about 4 rows back.
Thank you for an interesting talk. When the L forms divide into a variety of different sizes of daughter cells, what's the DNA distribution? Are the large ones polyploid?
Yeah, that's a great question. For a long time, we really struggled with that question. I'm interested in chromosome segregation as well, as you probably guessed. It's extremely difficult in under those imaging conditions to actually track the cells in detail. So we haven't really figured out how to do that until very recently. And what we've been doing very recently is to use microfluidic systems. So it turns out that if you drop an L form into a channel, which is about the same size as a rod, if you drop the L form in there, so you kind of constrain them into about the right sized rod shape, they actually then grow remarkably well. And you can follow chromosome segregation quite nicely in those cells, and they segregate not as well as the wild type, but pretty well. So I think it's a geometric problem that they have when they're used to growing, as it were, in a straight line and segregating their chromosomes in a longitudinal axis, and when they become three-dimensional, whatever the mechanisms are that they use don't work very well.
Yes, just 2 rows behind you. Yeah, there. Good. Thanks.
Thanks very much. In the fascinating slide you showed of division every 20 minutes, presumably something must have been feeding these bacteria.
Mm-hmm. Yes.
Is that food that goes in through the walls?
Yeah, these are typical bacterial growth media. So it would commonly have sources of carbon and nitrogen, sulfur and trace elements, plus often a carbon source like glucose, a carbon and energy source like glucose, and often amino acids and vitamins. And there are classical microbiological culture media that you can use to grow bacteria rapidly under those conditions.
The walls are semi-permeable to those molecules?
Yeah, they're semi-permeable. They also have, a lot of bacteria have incredibly sophisticated uptake systems. So they have transport proteins that will grab particular kinds of amino acids and transport them into the cell.
But if there are no walls in the L form, presumably the access is that much faster.
It could be, yes. No, it's an interesting point. One of the things we can test actually using this system is the question about the extent to which the wall plays a role in as a barrier to, for example, small molecule uptake, but also to protein secretion, because proteins have to go out through the cell wall, so we can look to see what happens when there's no wall there.
Oh.
Thanks. Yes.
Thank you very much for your talk. I bring greetings from Nick Lane, who hasn't been here this evening, but he passes his best. Has anyone done anything on archaebacteria? in this line? Are there any L forms?
No, it's interesting. There's very little information. Sorry, the question was whether, I guess similar things apply to archaea, the other major group of bacteria. There's very little information on L forms of archaea. And it's quite an interesting question. I wrote a little review on this that was published in Open Biology, the Royal Society's open access journal a couple of years ago. The thing is, archaea have a completely different cell envelope structure. They don't have the peptidoglycan cell wall, so they use different strategies, different molecules to envelop their cytoplasm. So it seems possible that the two groups of organisms actually emerged separately from the primordial soup as a result of inventing different cell envelope structures. So the peptidoglycan wall was invented by what became the bacteria, and the other kinds of wall structures were invented by the archaea, and they went their own separate way. But very little known about the archaea.
Yep.
Thank you for a fascinating talk. I wanted to ask you, how similar or different are L forms to mycoplasma? Because I believe mycoplasmas once did have a cell wall, and then they sort of shed it.
Yeah,
in
that's
their
true.
evolution. So I just wondered.
Yeah, it's thought that mycoplasmas, which as you say don't have any cell wall at all, came from probably emerged from the same lineage as Bacillus and Clostridium, but so they're probably descended from bacteria that used to have a cell wall. And you can imagine that what happened was that there was a Clostridium. It became an L form by losing its wall, and it then found some kind of environmental advantage by doing that. And the thing about the mycoplasmas is if you look at most of them actually are tightly associated, intimately associated with a eukaryotic or a higher organism host. And so they've probably evolved, having lost their wall, they've probably then undergone a lot of evolution in a direction that's helped them to interact with their host cell, which has resulted in them being very evolutionary different from the L forms. So the L forms are very similar, only maybe 2 mutations from existing walled cells, whereas the mycoplasma are, have undergone 100 million years of evolution or something like that.
Yes. Do you see that?
I just wondered, you mentioned that you grew the cells under microfluidics, and that changed the regularity of the progeny. Have you also tried mechanically stimulating the growing medium while the cells are being grown? Because presumably in original environments like hydrothermal vents, there'd be a lot of mechanical stimulation of the
Yeah, that's a good idea, actually. I'm not sure we've really explored that. So the idea would be that if by analogy to the lipid vesicle experiments, if you agitate those, you actually can increase the rate of proliferation. So I don't think we've actually looked to see whether that would happen to the L forms. We should try that. So we can get a better rate of proliferation by agitating them.
Yes, last one. Now, there.
You liken the peptidoglycan to a string vest, I think. Do you think, like a string vest, there's only one layer? It's a sort of a surface, or is there anything in the radial direction as it would be on a bacterium?
Sure. It turns out that the gram-negative bacteria, that one of those major groups, it looks as if it's almost a single layer, so like the string vest. But in the gram-positive bacteria, you might have noticed from the electron micrographs, it's actually a very thick, much thicker layer of peptidoglycan. So it's like a series of layers. And I think at this stage, one of the big unsolved problems in bacterial cell biology, if you like, is understanding the ultrastructure of the cell wall, particularly in the gram-positive bacteria. So it's multi-layered in gram-positives, but probably a single layer in gram-negatives.
Okay, I have one more thing to do, and that is, well, 2 things. One, to thank you for a fantastic talk, and how you brought it so much into current research, I think was just wonderful. Thanks very much. And then I have to give you this 3 things. One, the medal, and I guess a scroll of some sort. Thank you very much.
Thank you.